Thank you for introducing me. Today, good afternoon. Today, I would like to share our work entitled "An Alternative Method for Extracting Plasmodium DNA for Malaria Diagnosis in Subclinical Infection." Okay. First of all, I would like to tell you about the situations of malaria in Thailand. As you see in this screen, in Thailand, we have the organization that work directly to control malaria transmission. They have efficacy monitoring activity. So you can see the decrease of malaria case and malaria death from 2000 to 2019, around 96% reduction of malaria case and 98% reduction of malaria deaths. So the brown color is the non-Thai patient and blue color is Thai patient. However, it has a decrease of malaria infection. Nowadays, malaria is still the health problem in Thailand, particularly in border area, Thai Myanmar, Thai Malaysia, and Thai Laos. So this is a report in this year. Totally 2,344 malaria cases have been reported, and most cases are Plasmodium virus, around 2,000. And then the second prevalence is Plasmodium norisei and Plasmodium falciparum respectively. And the top five high prevalence of malaria infection. Here is the top five province reported in Thailand. You can see that is Yala and Prachuap Khiri-Khan. Here's the top five provinces that have reported many cases of malaria. These provinces are in the southern part of Thailand, and Kanchanaburi, Ratchaburi is the province that has high case of malaria, located in the western part of Thailand. So because this location is covered with forest and hill, that is the habitat of Anopheles mosquito. So for diagnosis of malaria infection, there are two approaches: clinical diagnosis and laboratory diagnosis. For clinical diagnosis, the symptoms, most symptoms are non-specific symptoms. The clinician have to consider the history of traveling into the endemic area and have to confirm by lab test. Nowadays, microscopy is still the reference method to diagnose malaria infection, and rapid diagnostic test is widely used also to detect malaria infections. However, these two techniques are low sensitivity, around 100 parasites per microliter can detect. These two techniques very low sensitivity. So molecular techniques have been developed to diagnose in case of low parasitemia. So, but molecular technique is so expensive, so it's not widely used in general hospital. And DNA extraction is an important step for molecular diagnosis. And but most of molecular technique, they like to use commercial kit to extract DNA. So commercial kit is, I think everybody knows, it's so expensive. Even it's easy to handle, it's easy to prepare, but it's so expensive. And another protocol to extract DNA is phenylchloroform extraction. This we call conventional method. Phenylchloroform extractions also labor intensive and time consuming. So that's the reason that why we try to establish new protocol to extract DNA from Plasmodium. There is the alternative way we try to establish the simple protocol and cost effective and takes less time to prepare. And here is the our work that has been published already in 2018. We compare with another technique, commercial kit, and previously published paper reported by Wang and co-worker in 1994. You can see for commercial DNA extraction, it has many steps. In general, for extracted DNA consists of cell lysis, and then DNA precipitation, and then DNA purification. You can see there are many steps in commercial DNA extraction kit. And for the conventional PCR, conventional DNA extraction published from previously, they prepare solution by themselves for lysis buffer. After they lyse cells or cell lysis, and then they precipitate DNA using sodium iodide and isopropanol, and then purify by 70% ethanol. Also takes time also. And for the new method, you can see we use only TE buffer and proteinase K, just only these two chemical solution. We lyse red blood cell by sterile distilled water by vortex vigorously, and then we centrifuge. We incubate with proteinase K for 1 hour. Incubate with proteinase K for 1 hour at 56 degrees Celsius, and then we centrifuge, and then we can use supernatant as a DNA template. It's just easy to prepare. Here's the primer that we used in our study. Actually, we used two sets of primer for PCR reaction and for semi-nested PCR reaction. We want to which technique that we can couple with our protocol to increase the sensitivity. So we try to use both PCR and semi-nested PCR. So here is the primer, the basic primer for semi-nested PCR. It just remind some of you a little bit. Maybe most of you familiar already how to do PCR. I just told you about the some of participants may not familiar more about the PCR. PCR has 3 steps: denaturing, annealing, and extension. Annealing is the denature, double strand DNA into single strand, and then specific primer to the DNA template, and then extension produce many copies of DNA in exponential. So how about semi-nested PCR? Semi-nested PCR has 2 reactions. The first reaction use genus-specific primer, and then dilute for the first PCR product, and do the process, the second reaction by the species-specific primer. Now this is to decrease the non-specific reaction. So in our experiment, we use leftover specimen. We use EDTA whole blood, positive PV and PF by microscopy. And then we extract using 3 methods that I have mentioned already. Commercial kit and published protocol, previously published protocol, and our new protocol, and then do PCR. So here is the result after the PCR with our new protocol. Lane C is negative control. It have no DNA template. Then see both A picture and B picture. A picture is the result PCR product from PF positive blood samples, and B picture is the result of PV positive blood samples. Lane C is negative control. I have no DNA template. Lane 1 is the PCR result from DNA extracted by commercial kit and link to PCR result from DNA extracted from a previously published paper, and lane 3 is the result of PCR of a DNA product extracted from a new protocol. So we succeed to extract DNA with a new protocol. And for the study, we want to test the limit of detection number of parasites that we can detect by extracted with new protocol. So we dilute positive samples in serial dilution and then extract without new protocol, and do both PCR reaction and semi-nested PCR to compare how many parasite limit of detection number of parasite that we can detect by DNA extracted from a new protocol. So we process both PCR and semi-nested PCR. And here's the result of PCR product. You can see the limit of detection after doing PCR. The limit of detection number of parasites was 40 parasites per microliter. This is a limit of detection for PF sample. And for PV samples, the limit of detection was 35.2 parasites per microliter if we doing by PCR. And how about semi-nested PCR? For semi-nested PCR, we have known already semi-nested PCR is higher sensitivity than PCR. That's I have mentioned for the principle of semi-nested PCR. We use to set a primer, the first pair of primer for genus specific. You can see the DNA target size 787 base pair. This is a Plasmodium species. And for the second reaction, we use species specific primer. For Plasmodium for genus specific primer, you can see the limit of detection at 40 parasites per microliter. But for the second reaction for semi-nested PCR, you can see increased sensitivity. We can detect the number of parasites at 1.6 parasites per microliter. And this is PF samples by semi-nested PCR. And for PV samples, you can see the first PCR reaction. We can detect parasites at the number 35.2 parasites per microliter. And for the second reaction, increase the sensitivity, we can detect parasite at number of 1.4 parasites per microliter. This is the advantage of semi-nested PCR to decrease the sensitivity, and that is proof that our new protocol can couple with both PCR and semi-nested PCR. And to verify our method that whether can apply in the real situation to detect malaria infection in subclinical infection. We use 11 samples, but samples from migrant workers have no symptoms of malaria and had proof already they were negative by microscopy, and use these samples to extract with new protocol. And then we did semi-nested PCR to verify that whether our new protocol can use to detect subclinical persons who infect with Plasmodium species. And here's the result after we extract the samples from 11 migrant workers, and we process amplified by semi-nested PCR using specific primer for PV and PF in the same reaction. You can see the A picture. This is the band control because the first pairs of primer for gene specific is also amplify human gene. It's like a control gene to make sure that we can extract genomic DNA. So every time we extract DNA from blood sample using this primer, we have to see this band. This is a human gene, like a control band. And you can see there is no product PCR of Plasmodium species. Have no band. The size of target size is 787 base pair. There is no band of target gene for Plasmodium. But for the second reaction, we dilute the first PCR product and then did the second PCR using specific primer for PF and PV, and we can see the positive band for PF. So for 11 samples, we see positive only 2 samples. This is the target band for this is 499. This is a PV positive samples, and this is the target size around 395 base pair. So here's the positive band for PF. So we use the blood samples from migrant workers that have no any symptoms of malaria, and we have and test already by microscopy. Okay, to compare these 3 techniques, you can see for commercial kit DNA extraction, they take around 4 hours, and cost per test, cost per samples around $2.86. And for previous published gene extraction, takes around 2.5 hours and cost $1.86. And for new protocol, it takes time around 1.5 hours and use only $0.15. You can see our new protocol is cheapest compared with other 2 protocols. So we can conclude that this new protocol can be applied to extract DNA, Plasmodium DNA in case of low plasmia, coupled with both PCR, and if you want to highest sensitivity, can couple with semi-nested PCR. This is a protocol that we provide. We can apply for molecular diagnosis for Plasmodium infection in case of very low plasmia, or in case of subclinical infection, or can apply to use for follow-up the patient that have been treated, and the clinician want to make sure whether they have a complete treatment to make sure that they are cured completely. So this is the things that we try to establish the new protocol instead of using the commercial kit that so expensive, or conventional protocol that have to prepare chemical solutions, many solutions, and maybe take more time, and use high cost than our protocol. So we like to. This is the aim of our study, But the disadvantage of our technique, because the DNA extracted from new protocol is not purity, so it cannot keep for a long time. But we have experience to keep it for a month. We can still extract DNA, still extract DNA when we keep for a month at minus 20 degrees Celsius. But we not recommend for keep in a long time. I think it's suitable to extract DNA and use immediately. Use immediately. It is. I think it's the most suitable for after extraction and use immediately. Not recommend to keep for a long time because it's not purity for this technique. So I hope that this my talk will help you to prepare in order to use outdoor protocol. That's all my presentation. Thank you.
Thank you very much, Associated Professor Dr. Porntip Laummaunwai. Are there any questions from the floor? There is the question from Professor Wu. Nice work. Some questions. 1. Did you try the method only boiling sample without proteinase K digestion? Did you try to use real-time PCR to compare with the regular PCR and gel electrophoresis? Do you think short size of PCR product can improve sensitivity?
Professor, so there are several questions. I did not try. I did not try for boiling samples with proteinase K, but I had review about they use microwave also. I saw the paper that they for boiling I have not seen, but for extract by microwave I saw the paper that I use a microwave, and the limit of detection they reported about 5 parasites per microliter for microwave, Professor. But I have not tried about boiling samples. It's interesting. Okay. And did you try to use real time? For real time PCR, I have not tried also. But the aim of this study, I try to save cost, Professor. I try to save cost, so I use the instrument that can be found in most of the healthcare unit. For real-time PCR, I think just only the in the slide university, and not real-time PCR is available in general hospital, but most of hospital they have a PCR machine. So I like to establish new method to provide for the in general hospital. This is my our team. So I didn't try real-time PCR, Professor. And do you think short size of PCR product can improve? Yes, I agree with this. I agree with this. So, but I use this specific primer from previously published paper. So I follow them. But I also interested to design primer by ourselves to prepare for the short size of PCR product may help to increase or improve the sensitivity. Thank you, Professor, for your advice.
Thank you very much. Are there any questions? You can drop the questions in the chat box, or you can also raise your hand if you have questions. So if there is no more question, thank you very much, Associated Professor Porntip Laummaunwai. Thank you so much. So